I'm Kieran Meade. I'm an ag student originally, class of 2000. Graduated and moved to didn't go too far, went to St. Vincent's Hospital and worked on Campylobacter, the most common cause of food poisoning worldwide, I think I'm correct in saying. Campylobacter and Salmonella, really interesting pathogens, as you guys know, because carried in avian species without any pathology, but make us very sick. So we're very interested in the immunology underlying that boundary between pathogens and commensals. So moved from Vincent's to Trinity, and then from Trinity to Teagasc, and I've been running my own research group in Teagasc for the last 10 years. So I'm about 30 days back in UCD now, and in a new position, new academic position in the School of Ag. So I'm just going to give you a little introduction to what my research program is about, and it hopefully will enable us to down the road link to a few people in the institute. So hopefully there'll be some common areas of overlap. Can everyone hear me okay? Yeah.
Yep.
Great. Okay. So really I suppose the principles from my research are around one, health. We know from the World Health Organization, and there's other publications that say that 60% of infectious diseases originate in animals. And it always amazes me because, as I said, I lecture and I worked in Trinity, and I'm quite familiar with the capacity that we have nationally in human immunology. And when you compare that to the lack of expertise that we have, or the lack of people we have working in other species, like emmetailing in bats, for example, or a very few small number of people working in bovine immunology or in avian immunology, it always amazes me, because really I think what we need to do is have redouble our efforts to control infectious diseases where at source, and invariably that means behind the farm gate or in wildlife species. The other principle that guides me as well is that the single biggest driver of antimicrobial resistance is antimicrobial usage. And as we know, the agricultural industry uses a lot of antibiotics. HIPRA estimate about 100 tons of antibiotics are used in food producing animals annually in Ireland. And we know across the world, diseases like mastitis are responsible for blanket antibiotic treatments that contribute to the emergence of resistance. So my own feeling is that we cannot feed the world, we cannot meet the demand for food, or we cannot sustain agriculture into the future without a revolution in animal health. And this is part of my work plan and part of the new masters that we're setting up in the School of Agriculture, which was supposed to kick off in September 2021. So my research then, in terms of the big picture, is to build capacity in what we call non-model organism immunology. So really to focus our attention on where these infectious diseases originate, so at source. And that therefore gives me quite a broad remit. And this schematic here just shows what we can do about animal health. Obviously the first step is always biosecurity, and we've seen that with COVID-19, critically important, but not directly part of my research program. But in other areas, the other 5 points my research does encompass, from designing better nutrition for animals to the identification and characterization of potentially new antibiotics, to improving vaccine design in livestock, and using new technologies to improve diagnosis of disease. And then ultimately, we would love to be in a position to breed for disease resistance at some stage in the future. So I'm going to touch on 3 of those areas in just a small bit more detail. So the first is my research to date has really with Clean O'Farrelly, who's in Trinity. We've a close working relationship over the years, working on what are called originally called antimicrobial peptides, and that I'm sure a lot of people in the institute are familiar with. However, as more and more roles for these peptides emerged in modulation of the immune system, they became known as host defense peptides. So we're interested in these cationic peptides and the potential that they have to provide alternatives to antibiotics. The second area I'll just talk briefly about is programming for innate immunity. I'm sure from a human perspective, this is obviously well advanced from a research perspective, but very little done in livestock species in terms of programming for immunity. And I'm going to talk a bit about that, and I'm very interested in this, and particularly what I've come to in the last number of years is a role for vitamin D. And again, I'm aware that with the Tilda study and everything, that there's a lot of very advanced vitamin D research, both in the Institute and in other institutes in Ireland. So we can learn a lot from that in terms of our application to livestock. There's also some interesting studies that have come out that I don't know a huge amount about, but the role of antibiotics, maternal antibiotics, on how they can impact on neonatal immunity and the microbiome in the neonate. So I'm just starting to get interested in that. And then one of my big projects really is to measure the immune response. If we think about all the traits that we have in agriculture, the traits that are economically important, like milk production or meat production, in order for us to make genetic progress and select animals with superior traits, we need to be able to measure them. But we can't reliably measure the immune response. So I'm looking at ways to try and measure the immune response, and I'll talk a bit about that. So we started off as a postdoc in Vincent's. As I said, we're working in Campylobacter, and we discovered panels of novel using kind of a comparative immunology and an evolutionary biology approach. We discovered new host defense peptides in the chicken, and then in the last number of years we've used similar approaches to characterize these host defense peptides we'll discover them first in other livestock species. My focus has really been on cattle. And what's interesting from the cattle genome perspective is that there seems to be an expansion in the number of these host defense peptides in the genome. So we're interested in the selection pressures that have driven that expansion, why they're there, and then can we exploit them in order to use them as therapeutics, say for mastitis, for example, or potentially in other species, even in humans. But there's lots of I'm not a peptide chemist, or there's a lot of expertise needed there in terms of the translation of those peptides into effective therapeutics. So I've written a review there recently, published in Frontiers in Immunology, which is the role these defensins, a particular class of these host defense peptides, are expressed at mucosal surfaces across the body, and we think that they play a role in regulating the microbiome. When we started to characterize these, like, one of the most interesting things about science, I suppose, is that you don't know where it's going to lead you. And we originally thought that, being host defense peptides, that these peptides were going to play a predominantly immunological role. But when we looked at where some of these defensins were turned on or were expressed, we found that they were expressed in the epididymis. And then we went on to develop antibodies, because of course reagents are not freely available for these proteins in livestock. And we've shown that the defensins actually bind to sperm and enable sperm to swim through cervical mucus. So it's a nice example of, they're also antimicrobial, but they're also playing a role in fertility. So I kind of was a bit of a vitamin D skeptic, I have to say, originally. I didn't think that, well, first of all, not being a nutritionist, I thought that, the nutritional programs for livestock were pretty well developed at this stage. I didn't think there would be such an obvious chink in their armor as vitamin D. So I started to ask around in Teagasc, and I asked nutritionists, and people advised me, well, we supplement with vitamin D and our animals are out in the sun all the time, so we're not going to have a vitamin D problem in Ireland. So when I started digging a bit deeper, looking for some data, it turned out that nobody had actually measured vitamin D in Irish cattle. So I said, well, given the wealth of important research that's coming out in the human field, I said we should really look at vitamin D in cattle. And you can see from some of this data here that we generated that we don't really know what the optimal threshold for vitamin D, for circulating levels of vitamin D are in livestock. But I put in a red dashed line there, which is 30 nanograms per mil of inactive, this is the inactive form of vitamin D circulating in serum, which is what the human studies tend to take as being required for optimal immunity. And we see that the vast majority of our animals in the winter period are below this optimal threshold. So part A shows cows after they've calved. At 7 days after calving, the vast majority of the cows are below this threshold. As our cows in part B, you can see as the cows go out to grass, in May or June, their vitamin D levels climb and they're sufficient or high in vitamin D. But then as the animals come back, as daylight starts to wane and the animals are coming back indoors, their vitamin D levels start to fall again below this threshold. So it's interesting because it means two things. It means one, our cows are calving in a vitamin D deficient or vitamin D insufficient status. And two, the calves that are born to those cows, you can see in part C, those calves take 5 or 6 or 7 months to actually come up above that 30 nanograms per mil sufficiency threshold of vitamin D. So I was surprised by this. It does seem to me that our cattle are in a vitamin D insufficient, they have a vitamin D insufficient profile. So I've written a grant application with some partners for the USDA, which we're waiting to hear about, which is to supplement cattle with vitamin D and look at a range of different health outcomes. So we know from the meta-analysis published in humans that your vitamin D level is very relevant to your susceptibility to respiratory diseases, and we know respiratory diseases are a big cause of mortality in calves on Irish farms. We know TB is a big issue, and we know vitamin D is very important for TB. And we're also, I have a PhD student who's looking at the role of vitamin D in reproductive function. So adding vitamin D to sperm to see if we can improve artificial insemination. There's also an area that I haven't really explored at all, but that's relevant, I suppose, to the Institute, whereas, you know, if we can improve the level of vitamin D in animal products, well, then that could have a benefit for human diets. Okay, so the third area then is in measuring immunity. As I said, like immunity underpins every other development that we're interested in agriculture. You know, if you want to breed animals, essentially I look at animals now with their elite genetics, that they're essentially high-performing athletes, and we need to make sure that, you know, they have a disease-free life as possible in order to meet their genetic potential. But we can't, if you were to ask any farmer or ask any person really, how's your immune system? We can't routinely measure the immune system. So we're developing assays in order to measure immunity, and we have a simple syringe-based assay, which I'm about to publish, looking at the immune response and being able to profile the immune response to different pathogens. But ultimately what we would like to do is put different components of the immune response on a chip, on some sort of a chip, and measure the immune response in large numbers of animals in order to breed for disease resistance or immunocompetence. So I've been reading up on how you would go about designing these chips, and I came across this NutriChip idea that I don't know if anyone in the Institute is involved in, but if there is any expertise in the Institute, I'd be very interested in talking to people, where people are using this NutriChip, which is essentially kind of a cell culture on a chip assay that people are using to measure how inflammatory different ingredients or different foods are. So the functional analysis of foods and how they may influence inflammation. So that's really the summary. Sorry, that was the NutriChip there. That's the paper, lab on a chip there, NutriChip, nutrition analysis meets microfluidics. So I suppose the key challenges in the email that I got from Emma was just, you know, what are the key challenges for your research? And I suppose my focus starting out in UCD now is to apply to the SFI Frontiers Research Program to develop this, what I'm calling immunochip or iChip assay. We're involved in setting up a Marie Curie ITN on the host defense peptides to try and move the kind of peptide discovery forward into therapeutics in livestock. And my particular role now during the lockdown, I suppose, is I'm setting up this MSc in animal bioscience, which will hopefully link to vets to really, you know, develop the immunology in livestock species. And then with the vitamin D research and the immunochip, my aim is to focus on the translation for industry. We're getting industry partners involved and hopefully some co-funding to try and move that kind of basic or strategic discovery research forward into kind of application.